Before we dive into today's episode, I want to take a moment to honor the legendary figure you're about to hear from, Dr. David E.Nichols. Widely regarded as one of the world's leading experts on psychedelics, Dr. Nichols has spent over 5 decades exploring how molecular structures can unlock the deepest workings of the human mind. In 1993, he founded the Hefter Research Institute at a time when psychedelic research was frozen by stigma and politics, thus helping restart the modern era of psychedelic science. In addition to developing novel psychoactive substances at his lab at Purdue University, Dr. Nichols also synthesized psilocybin, DMT, LSD, and other compounds for research programs at leading institutions, including Rick Strassman's groundbreaking DMT study in the 90s. He served as the Robert C. and Charlotte P. Anderson Distinguished Chair in Pharmacology at Purdue University and continues his work today, still shaping the present and future of psychiatric medicine. Dr. Nichols has influenced an entire field. Without him, the psychedelic renaissance might never have happened. His legacy is not only scientific, it really goes beyond that, and it's hard to put into words. But it's about relieving suffering, reigniting hope, and challenging how we understand the brain, consciousness, and healing itself. It was a true honor to speak with him. Before I get into the podcast, I just wanted to define some of the terms that Dr. Nichols and I discuss throughout the podcast, because sometimes the conversation gets a little bit technical, and so I will use this opportunity to define some of the terms. So starting with psychedelic compounds, we can define these as substances that alter perception, mood, and cognition, often through the activation of specific serotonin receptors. And when we talk about psychedelics, and specifically classical psychedelics, we are talking about LSD, psilocybin, and mescaline, so mainly compounds that primarily act on the 5-HT2A receptor, so the serotonin 2A receptor. This is different from other compounds that sometimes get labeled as psychedelics, such as MDMA or ketamine, but MDMA or ketamine are not considered classical psychedelics, and some people label them as not being psychedelics at all. Additionally, when discussing psychedelics, we can divide them into three main categories: tryptamines, phenethylamines, and lysergamides. Another important term that I will define as it shows up during our conversation a lot is neuroplasticity. And so neuroplasticity is essentially the ability of the brain to reorganize itself, forming new connections in response to experience. It is believed that the therapeutic effect of psychedelics comes from this ability of these compounds to trigger these adaptive changes at the level of brain circuitry. Within this, we also discussed things such as synaptogenesis and dendritic growth. So in the context of our conversation, synaptogenesis and dendritic growth simply means an increase in connections between these neuronal cells, so forming new connections and pathways. We also talk a lot about receptors. So you can think of receptors like locks on brain cells, and the molecules that we are discussing, such as neurotransmitters like serotonin or dopamine, as the keys. And so if the shape of the molecule is just right, it attaches to these receptors and it unlocks certain effects in the brain. This is a very simple explanation, but that's more or less what is going on. And so overall, these are just some of the terms that we discuss throughout our conversation. If you have any doubts, please leave them in the comments and we'll make sure to answer them. And so without further ado, let's start our conversation. Welcome everyone to another episode in the Mind Body Science podcast. Today I have a very special guest, Dr. David E. Nichols. Thank you so much for joining the podcast. I'm really excited to talk with you. And I guess to start our conversation, I would like to ask you about the paradigm shift in psychedelic research. So psychedelics were often labeled as hallucinogens or psychomimetic compounds that were used to simulate psychosis. But today we see that they have a very strong therapeutic potential. And during history, we have a lot of contemplative traditions that use these compounds as something to gain insight. In the middle of this historical progress, we also had a lot of political scrutiny over research of these psychedelic compounds. Basically, what I wanted to ask you is how has our perception of psychedelics changed over time? And why do you think why do you think research regarding psychedelics was so suppressed and it was very hard to do research.
So research was actually suppressed starting with Nixon administration and the Controlled Substance Act, the so-called drug war. In the United States, at least, there were a lot of protesters protesting the Vietnam War, and Richard Nixon was really angry about that. And he thought, Ehrlichman, one of his aides, has been interviewed and said, we knew that these drugs weren't dangerous, but if we made them illegal, then the people who use them could be arrested and given criminal records, and that would keep them from voting. And so we were trying to take the voting rights away from these so-called hippies that were protesting the Vietnam War. Principally marijuana and LSD were the ones that they wanted to control. So they made very stiff requirements, created this whole schedule of substances where you schedule 1 through 5, depending on their perception of how dangerous they were and their abuse potential. So it really started with politics. It didn't start with any danger. There were many, many hundreds of papers that were published of people using LSD in various kinds of research scenarios. The problem with most of that early research prior to, say, 1970 was that they didn't do it according to modern standards for clinical research, that is placebo controls and randomization, all the things that we know have to go into a formal scientific clinical study. So most of the early work was just dismissed, even though there were very tempting hints of the value of these. But everything kind of stopped. The last study that was shut down, I think, was in around the mid-70s at Johns Hopkins. That was the last formal study that was shut down. And everything stopped there until really in the 1990s when we had a resurgence of interest. There were some changes in the Food and Drug Administration. A new individual came in who found all these studies that people had applied for permission to run with psychedelics, and they'd all been put on the shelf. And so there was a big meeting, and I was involved in that meeting as well. Rick Dobbin was involved in that meeting, where we had a discussion about, is there any reason why these kinds of substances should be illegal to study and so difficult to obtain? And there was a consensus at the FDA at that point in time that if the studies were done according to modern standards for clinical research, that it was okay to do them. So that was approval, and that allowed Rick Strassman to do his study with DMT, which I think he did in the 90s, early to mid 90s. But that was sort of the beginning of it. And then I started the Hefter Research Institute in 1993 to get money to fund clinical studies, because the government at that point, they had been branded as being too dangerous to study, and the government wasn't funding research. They wanted to shut all research down. So I started the Hefter Research Institute. And over the years, we raised about $10 million from philanthropists. That allowed us to start to fund some of the early studies on psychedelics that were published 2011, 2016, in that era. And that really demonstrated that it was possible to do the studies again. You just needed financial support to do the studies. And of course, clinical studies are very expensive. The study that Michael Bogacic did at New York University in alcohol use disorder, psilocybin therapy for alcohol use disorder, I think we spent about $3 million on that study. So they were difficult to get the money and very expensive, but we demonstrated that you could do the studies again. So in the mid-2016, I would say, 2 studies from Johns Hopkins and New York University on psilocybin therapy for end-of-life patients. Those studies, I think, were really kind of a turning point for clinical research with these substances. But it was politics to start with, and then the recognition that these drugs could be valuable, and raising money from philanthropists all kind of got the studies started again in 2016, 17. And then there were studies in Imperial College that were also done about the same time with Robin Carhart Harris and David Nutt, looking also at psilocybin, doing some brain scanning. So everything started to ramp up again between 2000 and 2020, and at that point in time. And if you go to the literature, the published scientific literature, you see there was a lot of publications, and then he dropped off in around 1970. They started dropping off, and then about, again, that midpoint between 2000 and 2020, they started picking up again. And now there are a huge number of publications. Just the other day, I checked to see, use the word psychedelic, and there's a site that the government over here lists all the approved clinical studies, clinicaltrials.gov. If you go check there the other day, I found 590 some studies that had been approved by the Food and Drug Administration. So it's unbelievable. It's just taken off like crazy. And of course, now we know that they're very useful in treating depression. We had a major company in Europe. Now it's called Atai. I think Atai bought the company. But you have Atai. We have a couple of companies here that are doing work with depression. So it's taken back off again. So we had this 50-year hiatus, which is really due to politics more than anything else. It's kind of a shame because a lot of these treatments could have been developed earlier if they had been easier to work with and if there had been some funding.
Yeah, no, absolutely. And so, how can we formally define psychedelic compounds and what makes them different from other psychoactive compounds?
If we go to the pharmacology of these compounds, psychedelics, formal classical psychedelics, are drugs that activate a type of brain receptor, well, body receptor, but in the brain, known as the serotonin 5-HT2A receptor. And that is a very ancient receptor. The serotonin 2 receptor goes way back in evolution to really the single cell organisms. And, nature, when nature finds a genetic modification that gives a species an improvement, it tends to keep it from then on. And so this 5-HT2 receptor evidently is very important because it's been kept throughout history. And of course, it's one of the important receptors in the brain. It regulates emotional feelings, mood, lots of different things, appetite. And it's located on in the prefrontal cortex on the apical dendrites of cortical pyramidal cells, which are sort of major computational units in the brain. If you think of the brain as like a big biocomputer, the central processor would be these pyramidal cells, and they're activated by stimulation of serotonin 2A receptors. So they're really a key receptor. Now, people get into, is cannabis a psychedelic? There are people that will argue, it's a psychedelic. MDMA, is it a psychedelic? Well, some people say it is. How about ketamine? Well, some people say it's a psychedelic. They have different mechanisms of action. But the classical psychedelics, like LSD, mescaline, psilocybin, their targets are these serotonin 2A receptors. And they're the ones that I focus most of my research on. And there's a lot of research now to understand how the activation of those receptors actually produces therapeutic effects.
Why do you think these compounds are so promising for the field of biological psychiatry? I think psychiatry, in comparison to some other fields, I think there's a real need for new therapeutic agents. So, where do you see the promise?
Yeah, psychiatry is probably one of the most conservative, I'll use the word backward, maybe that's not the best word, but one of the most conservative areas of medicine. There haven't been new drugs. If you think about new drugs for psychiatry, we had chlorpromazine as an antipsychotic drug, which was developed in 1952. We had Valium and the benzodiazepines in the 1960s. And then you had the SSRIs like Prozac and citalopram, et cetera. And those all came much later. And those are the most recent additions, probably.
Yeah.
And they're not very effective. The SSRIs really blunt that effect. If you're depressed, they just blunt your feelings. They don't really cure the depression. So we've had all these fairly serious conditions that handicap people in their everyday lives. We haven't had good treatments, and now the finding that psychedelics are very useful. One treatment with a psychedelic can be more effective than weeks and weeks of treatment with something like citalopram. So these are being developed. I think we will see them brought forward as approved medications in the near future. Biggest problem is that the duration of action has been a hindrance. So you have, psilocybin, magic mushrooms still last 4 to 6 hours, LSD 10 to 12 hours, mescaline 10 to 12 hours. So if you think about doing therapy, no one really wants to. Well, first of all, they don't want to fund a therapy where you have to pay for some doctor or somebody to sit there for 6 or 10 hours with somebody. So the cost is prohibitive. So now you have places like Reunion, for example, is one of the new startups that develop psychedelics. And they're developing a modification of 4-hydroxydietyl because it lasts only about 2 hours. So shortness is a key thing everybody's looking at now. And you see a lot of people talking about what I would call non-psychedelic psychedelics. Delics as therapeutics is trying to develop these drugs that don't produce the change in consciousness, but alleviate depression. And there are maybe some uses for that, but people have asked me, what do you think about that? And, I can opine on that if you want me
Of
to.
course, yes, go ahead.
Yeah. Well, if you think about what they're using these for, if you have a major depressive disorder, just everyday run-of-the-mill, so to speak, depression, if you just take a pill and it could cure that depression, I think that's perfectly fine. And that's what companies like Delics are trying to do, finding this different mechanism, how psychedelics produce the antidepressant effect, and developing drugs that do that without producing the intoxication, the so-called intoxication. But I think in other conditions where you have people at end of life, I've seen interviews with people that have been given psilocybin who are cancer patients who are dying. And they're in an existential crisis, and it's not like major depressive disorder. This existential crisis when you're dying is a very different thing. It's really the most horrible kind of experiences people can have. And so when you take a psychedelic, it takes away, first of all, for many people, it takes away the fear of dying. And we're not sure how that happens. But they spend time looking at their life. And so, for example, I saw a video of one fellow in New York who'd been treated with psilocybin. He was a cancer patient. And he'd been estranged from one of his brothers, for years, 10 years. And after the treatment, he realized, I really want to reconnect with my brother, my family, and the people that I love and connect because I want the time that I have left to be really important and meaningful. And a woman who was treated in the first psilocybin treatment of alcohol use disorder by Mike Bogenschutz at University of New Mexico, and her video, she tried to get her husband enrolled because he was a heavy drinker, and he refused to be enrolled. And so she went to see Dr. Bogenschutz and said, well, my husband, he doesn't want to be in the trial. So he asked me, he said, well, do you qualify? Well, I don't know. So they had a questionnaire. She filled it out. And he said, well, you qualify for the treatment. Mm-hmm. So she took the treatment. And so prior to the treatment, she was a Hispanic woman who worked very hard. And I don't even know, she may have had more than one job, but she was a very hardworking woman and had several children, if I remember correctly. And she'd come home at the end of the day and she would just reach for the bottle of something to drink. And the kids would come around and she would just push them away because she wanted to drink and just wash away the troubles of the day, I guess. And after she took the treatment, she realized she was cheating her children of their mother. And after the treatment, she'd come home and want all the kids to come, and she'd put her arms around them and say, tell me about your day. What happened in school? Tell me about your day. So it completely changed the dynamics of her family by realizing the importance of her family as opposed to just drinking. So psychedelic in these conditions, either for drug use disorders, say cocaine, alcohol, smoking, it changes your perception. It's not you just don't stop doing whatever you're doing. It changes the way you're approaching life. And for people that are dying, it changes their perception as well. They want to reconnect with their family and friends. They want to make the most of the time they have left. A woman that was treated in Charlie Grove's study in 2011, she had a melanoma, a malignant melanoma, that the doctor said failed to resect the whole tumor. So it was still part of the tumor there. And it, of course, grew back. And she got into the therapy and she was very depressed. She had a 6-month prognosis to live. And so she thought, well, I'm dying. And she's very depressed, didn't want to do anything. She took the therapy and changed her attitude completely. She said, I'm not dead yet. I have life to live. And she reconnected with her husband. They started traveling, eating and going out to different restaurants. So she lived 2 years and then suddenly died of an opportunistic infection. But the quality of her life for those 2 years was dramatically improved over what it had been when she was just suffering. So you have these changes that there's a cognitive component for that kind of, for existential distress, depression, in drug use disorders. You see, I'm ruining my life. I'm ruining my family's life. Why am I doing this? And with the psychedelic, you look at that, it becomes very introspective, where the process, you say, why am I doing that? Why do I keep doing it? I know this is bad for me. I know this is bad for my family, but why can't I stop? And so there's a lot of introspection and personal analysis that happens. And so in those situations, I don't think you can do away with the so-called intoxication. I think that's an important part of it, understanding what your life has been, what your life could be, why you use substances. You're dying, okay, you're not dead yet. Let's make the most of the time you have left, reach out to the people who love you. So I think just for general depressive disorder, maybe general anxiety disorders, just a pill, if you can get one that works, I think no problem with that. But I think there are those conditions with substance use disorder, other kinds of disorders, where the introspective cognitive component is really important. And I don't think that taking away the intoxication, well, I use the word intoxication broadly, but I don't think taking the intoxication away will be the way to go with those. So I think there's really two ways to go, either just major depressive disorders or anxiety disorders where you don't know what's wrong and you just want to feel better. And then there's other conditions where you've been substance using, cocaine use, alcohol use, whatever, smoking.
Yeah.
It's a different situation. So I think that the intoxication there serves an important purpose.
And so in these positive situations that you show that psychedelic therapy can have positive outcomes, we can see these positive outcomes through changes in behavior and well-being and reported improvements in these people's subjective experience. But oftentimes we hear this term of neuroplasticity, and as we know, neuroplasticity can be adaptive or maladaptive. In the cases of psychedelic therapy, do we have some neural correlates where we can say and look in someone's brain and say, okay, after this psychedelic therapy, we can understand what the adaptive changes at the level of the brain have been?
That's certainly the goal. In cultured neurons, psychedelics, the activation of serotonin 2A receptors can cause neurons to sprout new dendrites. So dendritogenesis, synaptogenesis. In vitro and in mice, you can see that. I don't know that we've really figured out how to do that in humans. Obviously, there would be some kind of a imaging technique that could be used. I don't know of the studies where they've done that. You would think, for example, you might see resprouting of dendrites and neurons in the hippocampus, for example, I suspect that's happening. Most of the theories that I see that people have relate to neurogenesis really focus on that, the activation of, say, there's a receptor called a TrkB receptor, and that the activation of the 2A receptor leads to downstream events that stimulate the regrowth of synapses. I guess we know in humans that are depressed, you really have the pruning back of dendrites and synapses in the hippocampus. So they've done studies where they've shown, I think, where you give something like an SSRI, you see a resprouting and regrowth there. Well, I think the consensus seems to be that we're having, when we give a psychedelic, that we're having a regrowth of dendrites and synapses in various parts of the brain. Now, certainly they see it in vitro and see it in animal models. Presumably that happens in humans. So I think that's what people are trying to figure out, what it is, what's the sequence of events that does that. Because it actually, the therapeutic effects can occur quite quickly. Beckley now, or I think Atai's bought it, they're using an oral snuff form of 5-methoxy-DMT, which has been called the God molecule. It's extremely powerful psychedelic. People have amazing experiences on that. They only last about 20 minutes. And so you have people insufflate, their 5-methoxy-DMT benzoate, which is a salt that would insufflate it in their nose, and they have this experience that lasts an hour, maybe 2 hours with all the integration afterward, and the depression is gone. So it's hard to imagine that neuronal adaptation, sprouting dendrogenesis. It's hard to imagine that can happen that
Yeah,
quickly. But yeah, I don't know. I don't really know how quickly it can happen, but it seems pretty
yeah.
fast for me. So, but people think that's what's happening. So,
It's fascinating stuff. Nowadays, we have a lot of more human clinical trials in psychedelic research, but there's still a lot of animal experimentation. And so I wanted to ask you if what are the pros and cons or the value of animal experimentation when it comes to psychedelic compounds? Of course, we know that studying the subjective component of psychedelics is very hard in animals, but what is some of the value that we can still get from animal studies and psychedelics?
well, mechanism of action. For example, I did a lot of work with two-level
Mhm.
drug discrimination in rats. We trained rats to discriminate an injection of LSD versus placebo. Well, so we have two levers in this cage, and if you give them LSD, they press the right lever, and they get placebo and press the left lever. And you half the animals, then you switch half to LSD on the left lever, the other half on the placebo on the right lever. That was mechanistic study. So, for example, we made some LSD analogs that had never been tested in humans, and so we made an ethyl derivative of LSD we called ethylad, a propyl derivative we called prolad, allele we called allad, just pretty, but they were derivatives of LSD. Turns out in rats they're much more potent than LSD itself. Well, ultimately, some people took that, and some of the compounds we made we show were active actually came out on the black market as recreational substances. We had done studies, and so they usually the animal behavioral studies complement in vitro studies. So, for example, we did an experiment back around 2002. We published it where we took the diethyl groups of LSD. It was known that lysergic acid diethylamide, that those two ethyl groups were critical. If you had the dimethylamide, the compound wasn't very active at all. The methyl ethyl, it was kind of some activity. And then if you went to dipropyl, it lost its activity. So we thought, what is it about the diethyl that makes it important? So what we did is we locked those ethyl groups, which can move around and flop around freely, and we made some derivatives called azotidine. So they're four-membered rings with a nitrogen, and then we added a methyl on two sides. And so really what it was is a way to lock the diethyl groups into different shapes, if you will, or conformations. So we could lock them so that they were the two ethyl groups were sort of pointing in the same direction or pointing in opposite directions one way, and flipped over mirror image in opposite directions the other way. And we tested those in receptors and found that there was one particular orientation where the two methyl groups are in what was called the SS configuration, a specific orientation that was as active as LSD. So we said, oh, this must be the way that the ethyl groups are oriented. And then when we did in vitro studies in test tubes, basically in receptors, we also showed that that SS was the one that was active. So we published that paper in 2002. Well, in 2017, I think I was working in Brian Ross Lab, and we had the first structure of LSD in a receptor, an X-ray crystal structure of LSD in the two serotonin 2B receptor, which is similar to the 2A receptor. And when we actually looked at the shape of LSD in the receptor, those ethyl groups are exactly in the orientation that we had predicted from our studies. So the animal studies have been important to sort of draw correlations with what we see in humans. Mice are not really good translations for humans, but they're cheap. So people use this assay called the mouse head twitch assay. So if you give a psychedelic to a mouse, it shakes its head, mouse head twitch. And so the number of head twitches over a period of time correlates generally with the potency. And so it's a proxy for human activity. So that's one example where you can quickly screen a bunch of molecules to see, do they produce this mouse head twitch? And you can predict whether they might be psychedelic in humans. The other aspect of that, the FDA now has come out, and the government's come out and said, we're not going to support animal studies anymore. And the problem with that is, do you want to be the first person to be given a drug that's never been tested in another organism? And I personally would prefer that they test it on dogs and rats and find out it's not very toxic before they give it to me. So the animal tests are important for safety reasons to assure that they're safe before you might give a drug to humans. But basically, you can do correlative studies. Like I said, the mouse head twitch. Um, I'm with a company now called 2A Biosciences. We're developing psychedelics as anti-inflammatory agents. We have a library of compounds, and so one of the things we were looking for, if you want to give somebody a drug to suppress their inflammation, they may not want to get high, right? You know, inflammatory condition. Just take the inflammation away. I don't necessarily want to have a
Yeah.
trip. And so you can use that mouse head twitch as a proxy to predict, okay, we've done all these other assays that show it's an anti-inflammatory, then we give it to mice and it doesn't produce a head twitch. That means if we give it to a human, we probably are not going to get an intoxication. So I think animal experiments, they have various uses. They correlate with our in vitro test. They give us a prediction, at least in rats, potentially in dogs, although they're not used very much, give us a prediction as to whether a molecule might be active in humans and what level of activity it might have.
In terms of experimentation, do you think it is valuable for someone who is in the field of psychedelic science to directly experiment with these compounds? It doesn't need to be Alexander Shulgin style, but do you think there is some value here, or one can dispense with the direct experience?
The issue is really um, a moral issue that's been impressed on us by basically authorities. Saccharin was discovered accidentally. Somebody got some in their mouth and said, "Wow, that stuff is sweet." Um, and of course, Shulgin tested lots of compounds, and his data are used if you have animal experiments or in vitro experiments. You go back to see, well, was this compound active, and when Shulgin tested it. So what he did was very important to the field. He gave us a database of compounds which were active or inactive, and a lot of the compounds I sent to Sasha. Um, so we have a lot of ideas about what's active and what's not active based on experiments he did. But nowadays, it's frowned upon. So if you're an academic professor somewhere and it's known that you're taking, psychedelics, you might lose your job because of the moral imperative. It's morally a bad thing. So I think there probably are investigators who take these substances. They don't publish it because they just wonder, well, what is this compound like? But it's controversial because it's really a moral issue. I don't, if it's done properly, I don't think it's dangerous. Shulgin started with really low doses, in some cases extremely low doses. If he had a new chemotype that he didn't know anything about, he would start literally in the microgram levels and work his way up to where, he would either pick up an effect or it would have no effect, or he would feel like maybe it was affecting his heart and he would stop at that dose. So I think if human studies are done properly and you're really cautious about the doses, there's nothing inherently evil about that, but it's frowned upon. So the societal mores discourage it. And of course, your job could be, if you were a professor and you were doing that, you would endanger your position if it was known that you were experimenting with even new psychedelics that are not illegal. Of course, LSD, mescaline, psilocybin, and they're already illegal, so it would be against the law to use them. But if you came up with new molecules where there's no legality, it wouldn't be illegal. But then you have a societal mores that would say, no, this is bad. You shouldn't do this. So from a scientific point of view, I don't think there's anything wrong with it. But then it's generally frowned upon. So people think you're being very foolish or whatever.
Yeah. So, and in terms of the safety profile of these compounds, there are always concerns regarding neurotoxicity. I think for MDMA, there is some literature showing that there are some neurotoxic elements, although we can argue that MDMA isn't necessarily a classic psychedelic. But for compounds such as LSD and psilocybin, what is the overall safety profile when we discuss psychological safety, cardiovascular safety, and neurotoxicity?
From a physiological standpoint, Psychedelics, the classic psychedelics, are
Mhm.
very safe. If something like 30 million people have taken, estimate 30 million people have taken LSD, and there haven't been any fatalities. Um, Physiological. Now, psychosis is psychological, of course. You could precipitate psychosis. Um, some people are um, on the verge of developing some kind of a mental illness, and then they take something like high dose of LSD, and it can push them over into actual psychosis. So you have the possibility of someone who is predisposed to developing a mental illness being triggered into that full blown illness by taking a psychedelic. Um, for clinical studies where they're done in an appropriate set and setting, studies have been done with LSD and psilocybin. There's no evidence that there's any problem, that they're very safe drugs. Now, some of the newer ones, some of the N-benzyl, what we call the N-bones, those actually were physiologically toxic and killed some people. But for the classic psychedelics, LSD, DMT, mescaline, etc., physiological toxicity is not an issue for those. That receptor is not involved in any basic life functions. Just really the cognitive effects, potential psychosis would be the problems.
In terms of their potential for addiction, we generally say that psychedelics are non-addictive. Is this because they don't really modulate the dopamine system, or is it because there is some limbic friction in the experience, meaning that when you are having a psychedelic experience, there are things during that experience that you don't want to experience, so it's not all that pleasant, let's say? What are some reasons that may contribute to this?
Well, the drugs that are addictive either activate opioid receptors or produce an excess of dopamine. So cocaine or amphetamine used chronically, cocaine increases brain levels of dopamine and norepinephrine. The same thing for dextroamphetamine. All the psychostimulants, um, they modulate and stimulate brain activity. Um, and of course for the opioids, um, the mu opioid receptor, morphine, pyridine, things like that, they produce very pleasurable feelings, and so people want to go back. It's kind of a pleasure and anxiety effect mixed in. So people that take opioids, they really, I had my wisdom teeth taken out when I was a graduate student. I had a lot of pain, and they gave me Demerol, meperidine. Love that stuff. You take that and the pain is gone, no worries. So people tend to gravitate toward that, and so they're very addictive. You don't have that. The serotonin 2A receptor does not seem to modulate pleasure like that. So people take it, even if they have a positive experience. It's typically not the kind of thing that says, for most people, that say, "Oh, I want to do that again. I want to do that again now." Sometimes, I mean, a short-acting thing like DMT or 5-methoxy DMT. I know there are people that have really said, "Oh, I really like that. I do that as much as I can." But in general, people don't seek that kind of a feeling. It's more anxiolytic, benzodiazepine type things. Benzodiazepines are also dependence producing. Of course, alcohol is dependence producing, numbs your feelings. So, but psychedelics don't usually numb your feelings. If anything, they exaggerate and magnify your feelings.
Generally, psychedelic compounds are contraindicated for people with psychotic spectrum disorders. But do you think there are some exceptions to that rule, and why might that be?
Yeah, I don't know. All the clinical trials that have been done, they've screened the people out for any kind, first, if they have relatives, first order relatives that have a psychosis or any kind of psychiatric disorder, they'll screen those
Mhm.
people out. Um, Now, it's possible. For example, there was a study that was published years ago by Vanderbilt Vanguard, um, where they treated a person with obsessive compulsive disorder with monthly LSD sessions. And that really led to complete remission from his OCD. And that story they published, it was a 10-year follow up with this individual. And he became very successful in business and everything he had suffered from um, extremely bad obsessive compulsive disorder. I mean, he hears somebody flush a toilet in the house and he had to go wash his hands. Really a bad OCD, his OCD. And those monthly treatments without any therapy, actually, he improved and got completely well. And when the treatment was over, he got a promotion and became, one of their international uh, representatives. And so in some conditions, well, you could say somebody with a condition like that. In fact, there's an OCD trial at Yale that's being done now. And I haven't seen the data, but I've heard that it's very successful. So there are psychiatric conditions. Eating disorder is another one where eating disorder is one of the most fatal
Yeah.
psychiatric conditions, mostly strikes young women. And there's a trial going on now for psilocybin therapy for eating disorders. So there's a psychiatric disorder, and in those cases, people may get better. But I think for things that are
Mhm.
more like schizophreniform, psychosis, I think people are staying away. Now, ultimately, it might, Stan Grof treated people with LSD who sounded like they had schizophrenia, and uh, and they recovered. So it's possible that someday we may find out that
Mhm.
for some of these people, a certain type of psychedelic may actually be therapeutic, but nobody's really looking at that now, and that would be a situation that would be very, very touchy to have a clinical trial where you deliberately enroll people that have schizophrenia or some kind of psychosis. But I think it's conceivable that some of those conditions might even yield to therapy with the right psychedelic and the right conditions. But we're not at that point where we're ready to
Yeah.
try things like that.
Yeah, there's definitely some risk to it, but yeah, I think a question that fascinates me, and I'm trying to turn it into much as I can into a scientific question, which is that age-old question of why does the mystic happily swim in the same waters as the psychotic drowns? And so, what do we know at the level of neurobiology where we can predict whether someone will gain insight from an experience or whether they will fall into delusional thinking? Is there something we know in this aspect?
No, I don't think
Okay.
so.
Yeah, it's
The brain is the brain's still
yeah.
largely a black box.
Yeah. No, it's something that has been fascinating me for such a long time, and it's tricky to actually get to a point where you can create an experiment. And where do we start, right, in trying to answer such a question?
You know, recently I had my longest experiment lasted for all my career, um, and let me tell you about this. So Daniel Friedman was one of the first clinicians who investigated LSD in humans. And I knew him. I had many conversations with him. And we wrote a book chapter. He wrote a book chapter where he said, for most people who take LSD, the first 4 or 5 hours is a picture show in the head and pretty nice trip. And then for a certain percentage of people, about at the midpoint of the trip, it gets very dark. They get ideas of reference and paranoid ideation. It turns to a really
Yeah.
bad trip. And he said, nobody understands why that is. And this is a big issue. And I thought, well, that's really interesting. What's going on? So then I started going back and looking at LSD. And LSD, of course, one of the main theories of psychosis is based on amphetamine psychosis, an overactivation of dopamine, too much dopamine in the brain. So um, I published a first paper in 1976 where I pointed out that the structure of LSD was very similar to the structure of a drug called apomorphine, which activates dopamine receptors and the same stereochemistry and the whole bit. And I pointed that out. And people at Eli Lilly wrote me a letter after that and said, we've come to the same conclusion that this LSD has this dopamine fragment. It stimulates dopamine receptors. So I started wondering, but not everybody has that. So I knew a fellow who took LSD. He said, oh, it doesn't happen to me. 10, 11 hours, the whole thing was really trippy and happy. It's wonderful. But other people you talk to, and they said, yeah, after 4 or 5 hours, it gets really weird. I start getting these weird ideas, and
Yeah.
basically like schizophrenia kind of ideas. Now, why is that? And I wondered, could it be a metabolite? Could LSD be a form of a metabolite? So then it's known that there's a metabolite of LSD called 13-hydroxy-LSD. It's a major metabolite. And I thought, maybe this stuff activates dopamine receptors. So you take LSD and it produces this metabolite. And after a couple hours, you build up enough of this metabolite. Maybe it's a dopamine metabolite. So I proposed that. So then we did some studies with rats. And here's where the rats came in really importantly. We treat the rats with LSD and we use drug discrimination. So we give them LSD and then we train them 30 minutes later to see and compare other drugs and see if they acted like LSD. And so I thought, what happens if we wait longer than an hour or an hour and a half? So we treated the rats with LSD and we waited till the serotonin effect had worn off. And then we tested them. And they respond, but they don't respond as if they can give it a serotonin agonist. They respond as if their dopamine system has been activated. And so then we used a bunch of different antagonists. We showed that the dopamine D4 receptor antagonists could block that delayed effect of LSD. So it looked like LSD had stimulated serotonin 2A receptors in the beginning, but then as time went on, it's shifted. So now the effect was mediated by dopamine D4 receptors. And we gave them injections of LSD or a compound called DOI, which is a psychedelic amphetamine. We waited 2 hours and we gave them amphetamine and the effect was amplified. So I thought, now this is interesting. You give LSD and so activates serotonin 2A receptors. And now you have a formation of this metabolite that could be a dopamine metabolite, but you're going to be sensitive to that because the serotonin activation of that system somehow makes it so that a dopamine drug has more of an effect. It synergizes. So I actually, I thought, oh, we should make that metabolite and test it and see. So we tried to make that metabolite. I had 2 different postdoctoral fellows, and there was a natural products chemist at Brandeis University, and he had published a paper with a short synthesis of lysergic acid. And so I was following that synthesis, and I had 2 different postdocs, and we couldn't reproduce it. And we finally concluded that he had a graduate student who had faked his data. And I had sent him a sample of authentic lysergic acid. So once he had that, he knew what the NMR, mass spec, he knew what they all looked like, so he could fake it. And so then I contacted his professor, and I said, we can't reproduce this work. Can you tell me what's going on? He said, well, he had a job before he'd finished his PhD and asked if he could leave and finish his research in absentia. And so I approved that. So the student had left and faked his data. So that was the point at which I retired. So now with this company I'm working with now, we're working with a chemist at Florida State University named Joel Smith, who has developed a really 6-step synthesis of lysergic acid. It's really nifty. And so he made some of this metabolite, this 13-hydroxy-LSD. And this hasn't been published, and we don't have good numbers yet. But the first test that we ran in D4 receptors was 100 times more potent than LSD. So LSD has effects at the dopamine D2 receptor, D4 receptor, in the single-digit nanomolar, let's say 5 nanomolar. At the D4 receptor, it's 4 picomolar. It's 100 times more potent at the D4 receptor. So now that sounds like you form this metabolite, which is a super potent metabolite. It's 100 times more potent than LSD. So you only have to produce like 1% of that as a metabolite, and it's going to be physiologically relevant. So that's a case where animals have helped us. Now we synthesize this, and we'll be coming out with that paper. It'll be a year before it gets published. But anyway, it turns out that my whole hypothesis was correct. This metabolite that's produced from LSD in the body is one of these metabolites. Is a really super potent dopamine metabolite, dopaminergic metabolite.
One of the questions that often goes through my mind, and I generally start at the 2-factor model of delusion, where an anomalous experience isn't sufficient to actually cause delusional thinking and belief formation. There needs to be some other trigger that happens in order for the person to actually believe that experience or form delusional thoughts. So yeah, sometimes I wonder what the neurobiology behind that is. I think that's fascinating what you were mentioning about how somewhere in the middle of the experience it actually shifts. There's an effective shift.
So So if your brain has been primed to be a super
Hmm.
sensitive to a dopamine agonist, and now this metabolite is a super potent, it's the most potent dopamine agonist anyone has ever discovered. Nobody's seen anything
Yeah,
that's got 4 picomolar affinity activation at the dopamine D4 receptor. And we don't know much about what the D4 receptor does. But then I laughed. I said, some of the atypical antipsychotics block serotonin 2A receptors
yeah,
and block dopamine D4 receptors. Now, if we activate both of them, have we got a pro-psychotic here?
yeah.
So, you know, I'm not sure many people
Right.
want to take this substance just to see. It would be an interesting experiment to do a clinical study.
It requires some courage for sure from the subjects. Yeah, now that we have all of this discussion where we're trying to engineer new psychedelic compounds, do you think that there's any advantage in developing a psychedelic compound that is a pure 2A agonist? Because as we know, tryptamines, for example, they also activate the 2C receptor, which suppresses to some extent the 2A um, function.
A former student of mine, we tried. I was really
Yeah,
interested in finding a pure 1A serotonin 2A agonist because everything's contaminated. Even for the phenethylamines, they're 2A, 2B, and 2C.
yeah,
So you don't have a pure 2A. Because the 2C works in opposition to the 2A. You activate the 2A receptor, and then you activate the 2C, it antagonizes the effect. So like in the mouse head twitch, you see an increase in head twitch, but then when you get to higher doses, it starts activating the 2C receptor, and then the head twitch drops off. So it antagonizes that. But but I'm aware of a work one of my former students named John McCorvey in Wisconsin has come up with a trick for making very selective 2A agonist. And that work hasn't been published yet. But that'll be interesting to see. You know, he took advantage of what was some structural biology to develop a structure that's devoid of 2B and 2C activity. So it'd be really interesting to see when that molecule, when he publishes and they start doing work on it.
do we have some intuitions on what might happen?
Well, I don't. I mean, I think it'll be. I
Yeah,
think it'll be psychedelic, but maybe some of the confusion that arises when
yeah.
I don't really know. We've never had a pure 2A agonist, but I'm guessing that it'll be largely like a typical psychedelic because that's the main thing, activate the 2A receptor. And unless you give a large dose where maybe the 2C comes in, the major advantage would be in chronic, if one of these turns out to be useful for
Yeah.
chronic conditions. Uh, you won't be activating the 2B receptor, which is an anti-target, would stimulate fibrotic growth in the heart. So safety wise, it might be very useful to have a pure 2A agonist that doesn't hit the 2B and 2C receptors.
I love those questions where we don't really have much intuitions and it's just like it's open terrain, right? We don't know what we're going to get. In terms of that, we've touched upon this a little bit earlier on, but do you think it is possible to have, for example, I'll specify a case, for example, in terms of major depressive disorder, do you think it is possible that we can create a psychedelic compound without the psychedelic experience, which is as efficient as a psychedelic with the experience?
That's the,
Yeah,
that's the issue we sort of touched on earlier. If you talk to people who have taken a psychedelic and you say if we're going to take that away, we're going to give you a drug, but it's not going to produce that effect. Generally people find the psychedelic experience very positive. Um, you find the occasional person who said, I hated it, I thought I was going crazy, whatever. But for most people, psychedelic experience is kind of is positive. They get insights into their life and life patterns, etc. So I don't know. I mean, obviously, from a practical point of view, having something that alleviates depression, doesn't produce the intoxication for normal, just everyday life, I think that's something that's
yeah,
desirable. I don't know if it's possible to do it though, because the 2A receptor, um, although we functional selectivity, different ligands activate the receptor in different ways. It's possible that somebody may develop a ligand that activates the receptor and makes it an antidepressant, but doesn't activate the signaling pathway that produces the intoxication. I mean, it's possible somebody could do that, and maybe it'll be active.
Yeah.
There's certainly a need for that, a practical need, um, but I think it's going to be difficult. I know Delix is trying to do that, trying to develop ligands that don't produce effect. In the 2A biosciences, the ligands that we've developed
Mm.
don't produce a mouse head twitch, and they still activate the receptor at very tiny, very small concentrations. They're anti-inflammatory. So there's obviously a way to do that that doesn't produce a presumably intoxication if we believe the mouse head twitch is a
Yeah.
proxy for human activity. So it's possible to do that. And we basically modified it so it probably doesn't put helix 6 into the same position of the receptor. But we don't know, we haven't been able to figure out what the signaling mechanism is.
I was wondering what are the philosophical implications of this, right? If we are able to develop a psychedelic compound that doesn't really have the psychedelic side, but it's as effective as the compound that produces this experience that we can term, for some people, it's a mystical experience or something like that. It's, yeah, what can we learn from the brain if this is true?
Yeah, I don't think it's going to produce a mystical experience as you get one. I don't think it'll do anything. I think its effects will be largely imperceptible
Right,
if those people get what they want. I don't think it's going to produce any insights or anything. It's just going to relieve depression,
right.
um, ideally. So if you want the mystical experience, you know, you know, years ago when I took psychedelics, I wasn't heavily into it, but partly what got me interested in going to this field. I thought that it should be every human's right to have a psychedelic experience because it was so the one that I had, a couple were so powerful that people should be allowed to do this once in their life. You should be able to apply someplace and go and say, I want to have a psychedelic experience because I think fundamentally it's one of those things that you would only experience once in your life. And people who have, in some of the Johns Hopkins studies, they had questionnaires, and people said that taking psilocybin was among the 5 most important life events they'd ever
Yeah.
had. And among those events were things like falling in love, having a child, and of that order of magnitude, and taking a psychedelic was among those most important personal experiences. I think people should be allowed to do that.
And we have a rich history of also culturally speaking of it being part of ancient civilizations as well.
Yeah, yeah. Well, look at the Eleusinian
Yeah,
mysteries.
it's fascinating.
Every year for 2,000 years, every Greek citizen who was anybody, any Greek we've ever read about did this. They went experience this at least once in their life, and every Greek adult was allowed to participate once in their life if they hadn't been convicted of a, capital crime. I mean, it was an event. We don't know anything about it, but
Yeah.
they had the telestron. They had this whole the place was done, the village of Eleusis, a special place with the tenants all night chanting, etc. They drank this brew called kaikia. We don't know what was in it, but it was extremely important for 2,000 years in Greek culture. I mean, that's a pretty powerful tradition.
It is.
And until it was profaned in 400 AD, I think, when people started taking the kaikion for weekend parties, right? And so that kind of ended it. But until then, I mean, imagine every great Greek philosopher, every Greek anybody had participated, and it was considered to be, it was important. It was secret. You weren't allowed to talk about it on pain of death. So I mean, you know,
Yeah.
it's hard to imagine anything that's would be that important in our culture today.
Yeah. No, it is. And for this is maybe more of a philosophical question, but what do you think is the epistemic value of these experiences that people have when they're taking psychedelic compounds? Some people have this after they experience these compounds, they tend to say this feels more real than reality itself, and sometimes they get certain insights and conclusions. Do you think there is some value in terms of the psychedelic experience, not necessarily only in terms of personal insights, but also in terms of other areas of knowledge?
Um, you know, the inventor of the, I'm drawing, unfortunately, I'm 80 years old and memory, so people's names are really bad. Um, got the Nobel Prize for the discovery of the DNA sequencing. He said he got the insight after taking LSD, insight of seeing the enzyme jumping along the DNA molecule and making changes. I think there are people you could talk to who have got insights from a psychedelic. Maybe they've had would have had ordinarily, but they had some mental block, and the psychedelic that takes it away, opens it up. Um, I think there are probably useful insights that people have gotten and would get from an experience like that. Probably the most powerful experience I had, and I wouldn't say it was anything that was useful. Um, when my first experience with LSD, when I was 21 or 22, I guess, I had my eyes closed and suddenly I was projected outside the known universe. I was out in what I'll call infinite nothingness, if you can understand what infinite nothingness might be. And in the middle of infinite nothingness, there was this glowing ball, which I recognized was our universe, the known universe. And it was in the middle of infinite nothingness, and had a kind of hum. And the hum was life, and I had this impression that the function of the universe was to serve as a womb for life. And that was a really powerful thing that I had. And I can't imagine infinite nothingness anymore. I mean, I can say it, but it was the actual experience of infinite nothingness. In the middle of infinite nothingness was this universe. So that was a very powerful thing. I still remember it, obviously. Um, I don't know if it did me any good, but it was, emotionally, it was very satisfying to say, "Oh my God, that's a powerful, powerful impression of it's why we're all here. It's what it's all about. It's a womb for life, whatever." And so you can have powerful experiences like that. I can't prove that that's what it is. I can't prove that's true, but at the time, it felt like it was true.
Has your research with psychedelics informed or changed the way you think about consciousness?
No, other than to really impress by me that we don't understand what consciousness is.
The hard problem remains. Yeah, yeah, um, yeah. I think you've been amazing with giving me your time here. So I don't want to keep you much longer. But to end this discussion, I wanted to ask you what are some advices that you give to researchers who are starting out their careers and wanting to contribute to the landscape of science.
Um, well, I mean, science is understanding life, period. So any, in all of the different sciences, no matter what you're doing, you're understanding what life and time is all about. For psychedelics, if they want to study psychedelics, it's going to take a real dedication because there's no money for funding this kind
Yeah.
of research yet, and that may change. I did it because I was convinced that psychedelics had great value, and no one else was doing it. And of course, I started the Hefter Institute to fund work. I made the DMT for Rick Strassman. I made the MDMA for Rick Doblin. I made the psilocybin for Johns Hopkins. I promoted as much as I could because I thought the field was really interesting. And people that get in this field, if it continues to blossom like it has, um, they're going to be it's going to be a very feeling of personal satisfaction. I'll say a lot of people, I think you live your life, you get old, and you don't think much about whether you left anything behind. You have your family and your friends, and that's your life. For me, I have my family and my friends, but then I look at all the work on psychedelics that I prompted and started, and I think I leave part of myself behind. It's very satisfying because nobody was really looking at psychedelics that much, and I helped to fund this work. I kept the field going. I encouraged people to get involved. I started the Hefter Institute. So for me personally, that's the contribution I've made. It feels really good. Um, and if people get into science and they get into an area where they can make a contribution that really helps humanity in some way. It's a really satisfying thing. And most scientists they don't get out there and toot their own horn very much. Well, I didn't toot my horn, and then I started getting invited to give seminars when psychedelics got popular. Never imagined that that would happen. I thought I would always just be, you know. A woman asked me about 10 years ago or so. It's been more than 10 years now. She said, "What do you see as the future for psychedelics?" And I said, "Well, someday, long after I'm dead, you'll be having a midlife crisis, and you'll go down the street and you'll see a shaman slash psychiatrist, and they'll give you a session with a psychedelic, and you get a new perspective on your life." He says, "Oh my God, you think you'll be dead by then? How depressing." I said, "Yeah, I probably will be, but as long as the vector's pointing in the right direction, that'll be okay." So, I was hoping things would go in the direction, and they've taken off like crazy. It's amazing. But it's very satisfying because not many people get to be involved in a
Yeah,
field that takes off like that and have made seminal contributions. Presumably, I've done a few things that have been important in this field.
you absolutely did.
Somebody might be remembered from that. So, in addition to the normal things that people have, getting in science and making discoveries that really help humanity that's really satisfying.
Well, Dr. Nichols, as a researcher, if I do 1/10 of what you've accomplished, I'll be very happy. And it's been an honor to have you here. And so thank you very much for being here with me.
Happy to speak with
Thank
you.
you.